Hi, good morning everyone, and welcome you all to my class. Today, we can discuss on the topic data management in clinical trials. First of all, we know what, why, and when of clinical trial. Basically, in simple terms, clinical trial is a process which distinguishes a drug from a poison. And in order to distinguish a drug from a poison, we need to have credible data which can support our particular hypothesis. And this particular data should be even given to the regulatory authorities so that we get sufficient approval for the usage of our drugs. So, most importantly, the quality of data generated plays an important role in the outcome of our study. So, this particular data is usually collected by a particular process which can be termed as clinical data management. In short, CDM is the process of collection, cleaning, management of subject data in compliance with the regulatory standards. That is the primary objective of this particular clinical data management is to provide high quality data. The word which I am going to specify is high quality data by keeping the number of errors and missing data as low as possible and gather maximum data for analysis. So first of all, before going deeply into the topic, we should know what is this particular high quality data. High quality data means it is absolutely accurate and suitable for statistical analysis. In the fifth year, you are also doing your project work. So at the end, what we need of this particular data is that we need to use this data for statistical analysis, and for this the particular data should be represented in the most accurate and suitable way, and this is the main aim for which we are doing the clinical trials also. Another thing is that high quality data should meet the protocol specified parameters and it should comply with the protocol requirements, and this implies that in case of any deviation of the data which is not being specified in the protocol, we may need to exclude the patient. We know in our previous classes, we know the cost of doing the clinical trials, the per cost of a patient. So at the end of the study, if we are excluding a patient, that means it's a loss, a huge loss for the company. And this loss should be avoided. And if we have high quality data, or if we have the process for recording this high quality data, we can avoid this particular loss. Another thing is missing data. That is, during the process, the particular researcher is not recording the data properly. So it is also another headache for the statistical team and the clinical researchers. So high quality data should have minimal or no misses. That is, I've repeated once more. First thing is that high quality data should be absolutely accurate. It should be suitable for statistical analysis. Third thing is that it should meet with the protocol specified parameters. Fourth thing is that the deviations should be minimal. And fifth thing is that there should not be any kind of missing data. Now, we may require many tools for this particular clinical data management. We know the clinical trial process are usually multi-center. It is done by a large pool of professionals, and many software tools are usually commonly used for data management. And this particular software tools are usually termed as clinical data management systems, or CDMS. This particular CDMS can be of two types, commercial, and also there are open source tools. Most of the clinical trials are being done with commercial softwares, and the common commercial softwares are Oracle Clinical, ClinTrial, Macro, Rave, and eClinical suites. All these softwares are expensive, very expensive. They need sophisticated IT technology. infrastructure to function, and also specified trained professionals also. So keeping this apart, if you are planning to do a trial, a small trial, there are some small open source tools also available, and this commonly includes Open Clinica, Open CDMS, TrialDB, and Fosco. These particular softwares are available free of cost, and almost they are as good as their commercial counterparts. Now, the regulations, guidelines, and standards for CDM. We know the clinical trials. There are a lot too much of regulations and lot too much of guidelines. For example, ICGCP E6 guidelines. The GCP guidelines, it's a universally acceptable standard for conducting clinical trial. If in case of India, we have the Schedule Y. Schedule Y is a regulation which all the persons doing trial in India should follow. Like with this particular, CDM also has certain guidelines and certain standards. And the most common standard is that if we are doing a US FDA study or multi-centered study, international study, mostly nowadays all the data management is in electronic format, or some part of the data management will be in electronic part. So almost universally they may follow a particular guideline or regulation, that is 21 CFR part 11. Again, this comes under US Food Drug Act, that is US FDA, and the CFR is usually termed as Code of Federal Regulation. It has several parts. So in the 21st CFR, which is dealing with the clinical trials, the part 11 is dealing with the clinical data management. And the main, or what you can say, the main point in this particular part 11 is that the adequate procedures and control should be put in place to ensure integrity, authenticity, and confidentiality of the data. That is 3 points. Part 11 is mainly specifying on 3 parts: integrity of the data, authenticity of the data, and confidentiality of the data. All the researchers or all the CDM management team should make sure that these 3 points are satisfied while doing any trial. Another thing is that the Society for Clinical Data Management, SCDM, they have published a guideline, that is Good Clinical Data Management Practices, or GCDMP. Again, this is a guideline. This provides standards for good practice within particular CDM. Now, what you mean by CDM? We now know that CDM is a process through which the data is being collected, is vetted, or it's being cleaned, it's recorded and reported in this particular clinical trial. So what happens in the CDM, or what is the CDM process? The CDM process, like a clinical trial, begins with the end in mind. I will take your particular projects as an example. You have the aim, you have the objectives. So aim and objectives means this particular aim and objectives will come in the last part as the results. So like the clinical trial itself, while doing or while designing the particular clinical data management process, we should have the end in the mind. That is, as the clinical trial is designed to answer the research question, the CDM process is designed to deliver an error-free, valid, and statistically sound database. That is, how to answer the question and how properly we can answer the question. This is usually provided by the CDM team. And in order to meet this objective, the CDM process starts early, even before the finalization of study protocol. How this happens is that there will be a review and finalization of study documents. This is the first step in CDM. What happens is that the protocol will be reviewed from a database designing perspective for clarity and consistency. That is, before even showing the protocol to some other person, internally, or before even the proper designing of the protocol, the methodology part, just the methodology part, will be shown to the CDM personnel. And here the CDM personnel will identify the data items to be collected and the frequency of collection with respect to visit schedule. That is, how much data to be collected in how many visits. For example, if you have to collect the data of blood pressure, the CDM personnel should put proper spaces for collection of the systolic data and diastolic data, and also the particular intervals should also be mentioned. For example, you know the WH standard for collection of hypertension, it is usually 3 times, which is taken around 5 to 10 minutes apart in a single visit. Otherwise, if you have to take the blood sugar values, if you have to collect the HbA1c values, how frequently the particular HbA1c values can be collected, it should also be identified by the senior personnel, and the particular proper spaces should be given for collection. The next thing is that the data fields should be clearly defined and should be consistent, and the type of data should be entered should be evident from the CRF. That is, while looking the CRF itself, any lay person should be able to understand what kind of data should be entered there. For example, a common example, if the weight has to be captured, we are just taking the case that weight will be captured to 2 decimal places. Extremely correct data, that is, we are correcting in 50.62 kilogram, that is 50 kilogram 620 milligrams. Likewise, if we are planning to collect in 2 decimal places, the data entry field should have 2 data boxes placed after the decimal. Also similarly, the units in which measurements have to be made should also be mentioned to the data field: milligram per deciliter, gram percentage. Likewise, all the units should be mentioned after the spaces given for writing the amount. The another point is that the CRF or case record form should be concise, it should be self-explanatory, and it should be user friendly. So after what you can say designing phase, then comes the data collection. We know when the data collection comes, the first patient has enrolled in the trial, the patient comes with a regular visit, that is study startup itself, his initial baseline values will be recorded. Again, next visit, his visit one details will be recorded. So all this will be collected, and this collection will usually be done in a CRF or a case record form. And as explained earlier, there are mainly 2 types of CRF. What were they? There can be paper CRF and they can be electronic CRF. Now, the traditional method, or the most common method, or most easiest method will be to employ the paper CRF. And what happens is that in paper CRF, the investigator will record or will fill up the things, and after that the CRF will be sent to data response teams, where they will enter the data from the paper CRF to the database. That's first option. Second option is the eCRF, that is electronic CRF. In this, what happens is that the investigator or a designee will be logging into the CDM system, and they will enter data directly to the site. So these 2 methods are there, either paper CRF or electronic CRF. One, we are entering the data at the site. From the site, the CRF will be taken to the data management team site. From there, they will enter to the database. Second method is that the investigator will enter the data directly to the database using proper login systems. Now, after CRF entry, the next portion comes is the CRF tracking. CRF tracking means the entries made in CRF will be monitored by the clinical research associate. Usually we have a term for this which can be termed as monitoring. So during the monitoring, what happens is that the clinical research associate will come to the site, he will be having the CRF, or he will take the CRF, either electronic or the paper, and he will compare it with the source documents. Why? In the sense to make sure that all the data are being recorded in the proper way, there are no missing data. Now, this clinical data management team will track the retrieved CRF and maintain the record. That is, only after the monitoring, only after the correction, the CRF will be given to the data management team, and the CRF is usually tracked for missing pages or illegible data. Again, in case of missing or illegible data, a clarification should be obtained from investigator and the issue should be resolved. That is, ultimately we are doing the clinical trial for getting data. If something is missing, that means loss. So during the trial process itself, if there is any case of missing or any case of eligible data, the data management team should make sure that the particular issue is resolved. Now, after the CRF tracking, there comes the point of data validation. Again, we have heard about this validation term. What is validation? Correctness. In simple terms, correctness. That is, what happens is that data validation is the process of testing the validity of the data or the correctness of the data in according to the protocol specification. So how this is being done is that there will be certain edit check programs. Edit check programs in the sense is that if in case there is any kind of discrepancies, that is any mistakes, automatically itself the system will give some check, edit check and edit. So this means the particular data should be rewritten. So the discrepancy is defined as a data point that fails to pass a validation check. That this is usually automatic. That is, there will be discrepancy. It can be inconsistent data, it can be missing data, it can be range checks, or it can be deviation from protocol. For example, if you take the inclusion criteria specified at age 18 to 65 years, both ages which should be written as less than 18 or greater than 65. If you are writing 16 years, automatically the software will give an edit check. Otherwise, we have particular we are recording the FBS values. FBS, you have a usual range. If it is abnormally higher FBS, somebody is writing 723 milligram per deciliter, automatically the particular program will give an edit check. That is a range. The range is not consistent. Now, once there is a data validation, next what we have is a discrepancy management. Discrepancy management is that we have identified a wrong information. Now, how to manage this particular wrong information? This is the discrepancy management. This particular term is also termed as query resolution. Query in the sense, when we see a missing data or eligible data, we will raise a query to the investigator, and this particular query should be answered so that the particular discrepancy is solved. Now, discrepancy management includes reviewing discrepancies, investigating the reason and resolving them with proper documentary proof. And what the main aim is that it will help in the cleaning of the data and gathers enough evidence for deviations observed in the data. Now, what generally happens in the particular study is that in the Cerium team is that whenever they identify a discrepancy, they will give a self-evident correction form with or without sending a data clarification form to the site. And two methods, that is, there can be either a self-evident correction, SEC, or there will be sending a data clarification form to the site. The most common self-evident correction is that no need of clarification from the investigator. This can commonly occur in case of spelling errors, minor spelling errors. In this case, the particular data management team, without giving any kind of clarification form, can clarify the matter. At the same time, if it is something more relevant, for example, the FBS value, RBS value, something of importance, then we have to give a data clarification form. And data clarification form will be given to the investigator. The particular investigator will correct the data and he will send back the data along with the clarification. That is why this particular error came. That is, the investigator will write the resolution or explain the circumstances that led to the discrepancy of data. Now, in case of ECRF, the investigator can access the discrepancy, flag to him, and will be able to provide resolutions online. And once a particular data resolutions is given, it will be recorded as closed. So thank you. I'll be continuing the next class in the next session.